I'm going to talk a little bit about phosphopeptides, and one thing that I've kind of left out of this is why are we going to look at phosphopeptides? And so I guess I assume that as the biologist, maybe that you guys know this better than I do. But it's I always used to like to say it's something you can't do with mRNA. Always kind of grew up scientifically competing against the genomics world, and so post-translational modifications are something that the proteomics and mass spectrometry world has said, oh, we can do this and you can't. But to be fair, it is more difficult than a normal proteomic experiment. But it is something that can be routinely now done with a mass spectrometry approach, looking at signaling. Phosphopeptides are fairly rare, so you're looking at maybe 1 to 5% of peptides may be phosphorylated, and so you do need something to pull them out of the mixture just because of abundance. And I'm not picking on this paper, but just showing that when you look at really any study, and they use different methods to enrich the phosphopeptides, they always see differences. And so in this, they use an iron IMAC versus a titanium oxide, and they see some phosphopeptides that overlap, some that are better for one, some are better for other. And so this is a little bit like if we had a show of hands and said, okay, which is better? Is a Ford better or a Chevy better? And so the answer, of course, is Chevy, because I grew up in a family that drove Chevys. Mike Kinter would say, well, it's obviously the Ford. Dennis would probably say, you're both wrong, it's Toyota, and so and it's kind of which camp you're from. I don't care. I am enrichment agnostic, let's say. So I just want to know which is going to be, which is going to give us the best data. Well, let's look at this a little bit. What are we doing? And so I can talk just very slightly on inorganic chemistry, because inorganic chemistry was the field that I did the worst in graduate school, and so my degree is in analytical chemistry. Anything physics related, I did well. For some reason, coordination chemistry and inorganic, I struggled. And so I think it's God's way of laughing at me that now I use coordination chemistry on an almost daily basis. And so, but really what we're doing with phosphopeptides are leveraging the fact that we can take advantage of this unique substance that's on a peptide. And so peptides get phosphorylated to change their activity, and they do this by changing charge. And so the phosphate changes the charge on the peptide or protein, which changes its conformation, either opening up or closing an enzymatic pocket to allow activity to turn on or off. And so we can use this charge effect as a way of putting a handhold on it. And so if we take a compound that we immobilize titanium to, or titanium oxide, we can then use the fact that this positively charged titanium is going to interact with the negatively charged phosphate, and it's going to grab hold of those. We can do the same thing if we use iron. You can use gallium. People have used, a range of things. And it works reasonably well. There's lots of literature on which is the best one, and it's this is a lot like chicken soup. Your mom's chicken soup is the best because that's the way she makes it. If you look at the ingredients, everybody's ingredients for chicken soup is the same, but everybody makes it a little different, and that's just kind of like these enrichments for phosphopeptides. All the protocols are essentially identical, but they're all pretty highly variable. And I challenge any of you to take a paper that gives a long protocol for doing phosphoenrichment and having two people in the lab do it and get results that are anywhere similar to one another. And so, but now, after years of doing this, there are at least a few studies that have kind of shined a light on some of the problems. And so, and this is something that I had observed and really not known why. I knew that if we did just a solution digest, if we just took a protein in a tube, did a digest, the phosphoenrichment wasn't as good as if we used the filter-aided method, where we use a 30 kilodalton filter to digest and spin the peptides through. I didn't know why. I would think that if anything, adding something in like a filter would make it worse. But in my hands, it was always better. In this study, one thing that they realized was if they added benzonase to chew up the nucleic acids, their recovery really went up on phosphopeptides. And so if we look, I've circled in red here, this is the standard protocol and this is the improved protocol. And first, as an analytical chemist, I look at this and go, okay, the peak's bigger here. When I first read this paper, I thought this is mislabeled, right? This has got to be the improved protocol. It's a bigger peak. What their point is, this little peak down here, these are the phosphopeptides. The big peak here is everything else that you're also enriching that's getting in the way. And so it's DNA, it's other charged things that are binding to this titanium and iron, and other charged things. It's not surprising that DNA and RNA, with phosphate backbone, is going to bind to something when you're trying to enrich phosphopeptides. You chew them up, get rid of them, your recovery is a lot better. And so this, kind of explains some of the variability that you see in lab to lab. I saw this poster at a mass spectrometry meeting from Thermo Fisher talking about a couple of kits, and I was highly skeptical because I'd used these kits 2 or 3 years before, and they showed, again, like everyone else, a difference in titanium versus iron, a difference in multiply phosphorylated versus singly phosphorylated. But in my hands, I'd used these before and they were just awful. I didn't get much of anything. I looked at the kits and then realized that Thermo has sold these kits for years. But a few years ago, they changed the buffers, same part number, they changed the buffers and now they actually work. And so it works really quite remarkably well. Titanium oxide does do a better job of binding more highly phosphorylated peptides, but sometimes it can be, if you have a lot of multiply charged or multiply phosphorylated peptides, those peptides will bind up all the resin, and the singly phosphorylated peptides will flow through and not be enriched. And so the way we get around this is that we take the peptides, we enrich them with titanium, we take that flow through and then enrich them with iron, and then combine them back together. And so we're not worried about which one is specific to one method or the other. We're enriching with both methods, putting them back in the same pot, and doing the analysis. And so we did this with a fairly small sample. These are NRK, normal rat kidney cells, 30 micrograms, which is really kind of a small protein amount for phosphopeptides, isolated with titanium oxide and iron. We actually ran these separately to look at the overlap, and it was indeed about what they said on the in their poster. But these were in 2-hour runs, so four hours of instrument time, and we identified over 13,000 phosphosites, and over 9,000 of these were quantified by TMT. One interesting thing is if we look at the distribution of phosphopeptides, so these are sites that are called by the software. 79% are serine, and this one 18% threonine, and 2% tyrosine. And this falls within about what you'd expect. So about 80 to 85% of phosphorylation occurs on serine, about 15% on threonine, and this is highly dependent on the cell, but 1 to 3%, or in some cases 0.1 to 3% are phosphotyrosine. Much lower abundance. You can see here, over 10,000 phosphoserines, 288 phosphotyrosines. So phosphotyrosine, I'm not really going to get into that here, but if you're interested in signal transduction in tyrosine signaling cascades, that has to be a separate enrichment, not just the generic titanium or iron. But this is, pretty quick method. You can get, 10,000 phosphosites. Well, what happens if you have a little bit more sample? So our phospho-TMT workflow is standard with 200 micrograms per sample. And we do this so that we can, use this bulk of material to enrich these lower abundance phosphopeptides. We digest, as Sam said, we label, we do high pH fractionation, and actually before we do the fractionation, we take 95% of the material to do a phosphopeptide enrichment, and we only take 5% and do the proteome analysis. These we typically fractionate in 12 to 40 fractions. So this is 12 to 40 hours on the mass spectrometer for a lysate, and for the phosphopeptides, again, the same. The depth depends on how much time you throw at it. We recently did a study where we're looking at it's actually a mouse tumor model, MDX model, where we're looking at human tissue grown in mice, melanoma, and in that we ran 34 fractions, so threw a lot of instrument time at it, and identified over 30,000 phosphosites. And so if we make this more stringent and say, okay, only show me the ones that are 90%, have a 90% probability of being localized to a single site, it's still over 21,000. And so, really good deep data. It's almost too deep. It's so much data that it's a little hard to deal with. Luckily, we have really smart people like Aaron Story, and he's developed some software along with the bioinformatics group, ProteoViz, that allows us to visualize this. And so now this software is being implemented in our reporting structure. But one of the things that this software allows you to do is look at this data with a volcano plot, and then we have a lot of information that normally you'd have to sift through, pages of Excel spreadsheets to get to. You can look at a certain spot on this volcano plot showing a high fold change and a significant p-value. You get the protein name, the peptide. You get the probability that phosphorylation is on one site versus another. And so in this case, there's a 78% chance that it's serine, 22% chance that the phosphorylation is on this threonine. And the software also allows you to take a comparison like this. So you look at a volcano plot, it's always a binary comparison. So you can take these phosphorylation changes in one system and see how those the abundance of those phosphopeptides change across all of the samples in your study. And so we can do that by generating heat map and look to see how samples cluster. And so it really gives being able to visualize the data like this gives a researcher some power. We can take these and again look at the motifs. So if we see a phosphorylation change, we can look for a motif and try to back out which kinase is there. Or in the same regard, if you're mutating a kinase and you see phosphorylation changes, you can know if it's that kinase that's making the change, or if it's a downstream effect based on the specificity of your phosphorylation changes. Finally, I just wanted to highlight this paper. If you're looking at phosphorylation data. The online databases really aren't very good, and so a lot of the phosphorylation data is still kind of linked at the gene level, and it's not at the specific site level. And so this is a paper out of the Broad Institute, and they've gone through and curated thousands of data points where they're looking at specific sites of phosphorylation change and how that affects downstream. And so we're using the database that they built here within ProteoViz to help the downstream processing. And this is something that our bioinformatics group and Aaron continue to work on. So, all right, threw a lot of phospho at you, but I can pause here and take any questions about phosphorylation or